My name is Emily, and my proposal is about investigating enzyme kinetics in microgravity. My research question is, how will exposure to microgravity impact the speed at which the enzyme lipase breaks down lipids? So my background, what do we already know about this topic and what research has already been done in this area? Lipase is an enzyme whose function is to break down lipids into things that can be easily absorbed by the body during digestion. If lipids can't be absorbed by the body, this causes significant impacts on vitamin bioavailability, gastrointestinal function, and other things. A lot of research has been done to try to figure out why the immune system is so impacted by microgravity and radiation exposure, but not much is known about the kinetics of specific enzymes in microgravity. So that is the gap that I'm trying to fill with my research. Why is this important? Why do we care? Studies have shown that exposure to microgravity has many significant impacts on the human body, its many complex systems, such as bone density, liver function, vision, digestion, etc. As technology advances, long-term spaceflight becomes more and more imminent. But for astronauts, this also means lots of exposure to microgravity and radiation. And the impacts of these things need to be understood in order to best protect our astronauts. So this is a diagram of my experimental design, which I'll go into more detail about on the next slide. But this essentially just shows how I intend to set up the FME kit for this experiment. And this is my experimental design written out. So basically 2 days before undock, the astronaut on the International Space Station will unclamp the first clamp and shake the FME, which will mix the lipase and the lipids together. In this experiment, I'm using extra virgin olive oil as my source of lipids and porcine pancreatin as well as bile salts as my source of lipase. For 100 seconds, these will be allowed to react, and then the second clamp will be released and hydrochloric acid will be introduced to the system, which will stop the reaction. This procedure will be replicated on Earth, and the pH of each sample will be taken in order to determine how lipase functions in microgravity compared to Earth. So what will the results of this experiment tell us? If we find that lipase function is depressed by microgravity, then astronauts may need supplemental lipase in order to keep them as healthy as possible. If no effect is determined, then altered digestive function in space must be due to something else that we haven't discovered yet. But whether or not an impact is determined, we will learn more about how exactly lipase and other similar enzymes perform their functions. And that's all I have. Thank you so much for your time. Are there any questions?